All right, welcome to Talk Session 2. We have a lot of great talks lined up for this afternoon. And we're going to get started now. So I am your first speaker. So my name is Kristen Davis. I am at the University of California, Irvine, which is a little bit up the road from here. And I am going to talk today about some work that we've been doing over the last 6 years or so on seaweed farms and actually trying to bring numerical modeling as a tool to kind of help do lots of things on seaweed farms. And was it audience? Okay, thanks. Okay, so the work I'm going to talk about today is not just my own. It is a large group of people, students, postdocs, farming partners. So this is a collaborative effort for sure. We were hearing just in the tent next door, if you saw any of the panel discussion about how tight-knit a group these, seaweed farmers and scientists are especially in California right now. We are really working side by side, which is fun. Makes it a lot of fun. So this model that I'm going to talk about that we've been using is called MacMods. It's the macroalgae cultivation modeling system. And we intended for it to be useful for a lot of different things. So everything from, trying to understand how to design a farm to optimize yield on farms, not only where we're used to farming seaweed, which is close to shore, but farther offshore, where there might be a little more space to put farms. And farther offshore, often there's not a lot of nutrients in the upper part of the water column. And so we wanted to use the model to think about ways we could design farms to potentially passively upwell water into the farm to feed the kelp. The other kinds of things we want to use the model for is to site farms. We have some information about about water temperature in the ocean, about nutrient concentrations in the ocean. We know about light fields, and so we can use that information to say, if you were going to seek a permit for a farm, where would you optimally want to look for it? You also need information about where there are other uses already, right? So you can't put it in the middle of a shipping channel, and you can't put it in the middle of a marine-protected area, but you might also want to put it close to port if you were going to harvest your yield and you wanted to get it back to port. So there's lots of different factors that go into choosing a site for a farm. And so the things that we're trying to do here range in scales, both in spatial scales from really large scales to really small scales, especially if you're trying to think about farm design. And so that's a real challenge for modeling because models, you kind of have to choose a scale and focus on those questions. So we actually accomplished that through kind of pairing some models together, a regional oceanographic model that gives us some information about the ocean state, a really fine, high-resolution hydrodynamic model that can actually resolve flows and mixing within the farm, and then a dynamic macroalgae growth model that gives us the growth component of our farm model. And this is published. I invite you guys to check it out. This is a paper led by a postdoc of mine, Christina Frieder, who's now at the Southern California Coastal Water Research Project. And it is also, the code for our model is on GitHub, open source. Feel free to explore. So one of the wonderful things about this project is that we've been able to work directly with some farm partners here in California. So up off of Santa Barbara, we've been working with Ocean Rainforest. Maybe you heard earlier from Eliza Harrison, who talked from Ocean Rainforest. We've been working with Loretta Robertson, who has Tropical Seaweed Farm Project in Puerto Rico. And we've been working with folks from Ocean Era off of Kona in Hawaii. And this is an example on the movie up there is an example of model output on one of the macrocystis farm simulations that we've been doing. So flow is going from left to right, and you're looking down on the farm, and it's kind of going through these lines of seaweed, of kelp that are planted along the lines. And what we can see is the flow is slowing down as it's dragging on the kelp and accelerating in between. And it drives some really interesting flow features. And in fact, it's driving some vertical eddies that are really interesting and potentially for passively upwelling water into the farm. And so we're using this model to really get optimizing farm design in order to feed the kelp and grow it better offshore. And of course, models are just, we solve the equations, we get a solution, but we don't know for sure if it's all right. So we actually need to go into the field and take measurements on farms. This is us in the field on the Ocean Rainforest Farm off of Santa Barbara, collecting measurements to kind of ground truth our model. And then we're using this model to do things like predict seed out dates. What is the optimal seed out date for macrocystis at this site to get the best yield over several years of different ocean conditions? Or what are the trade-offs between light and nutrients for different depths of cultivation lines? Or what kind of harvesting strategy would get you most biomass at the end of the year? And we're also our next steps are to really target some of the environmental and the environment farm feedback. So we've thought, you can kind of think about it. Okay, well, one farm in the Southern California Bights probably not going to make a big difference. But if we, sort of really built out seaweed farming in a big way, we would begin to affect things like circulation, nutrient cycling within the coastal ocean, shading of phytoplankton. So these are all things that we can use modeling tools to begin to get an understanding of those feedbacks. So that's where we're headed next. And we've been using our model output to build some tools for farmers to kind of think about site selection. So here is this green background is really our MacMods model output for yield in different places in the Southern California Bight. Darker green equals more kelp. And so really you can see that's even in deep water where, of course, natural kelp doesn't grow. But if you had a farm, you might be able to farm there. And then it's overlain with things like shipping channels and marine protected areas, places where you wouldn't be able to seek a permit. And so this is a sort of ArcGIS tool that some software engineering students at UCI helped me put together during a master's project recently. The model is also being used as part of the Department of Energy's billion-ton report, which is a report that they put out every few years to look at trying to estimate different sources of biomass feedstocks for energy, for biofuels. And this is the first year that they're going to be putting a macroalgae chapter in. And of course, there's not a lot of macroalgae being used in biofuels yet. But what they want is some projection of the possibilities. And so we're helping them to get those projections with our model. And of course, we've been hearing a lot, if you've been listening to things about concerning seaweed, that a lot of people are really excited for the possibility of sequestering carbon dioxide out of the atmosphere by soaking it up in the upper ocean through seaweed productivity. And right now we are trying to use our model to look at that question as well. We sort of started out by there's a lot of really complex things to explore in this problem. And I think something that was said in the tent, if you were sitting over in the next tent just a few minutes ago, is that we have a long way to go to answer this question. I agree. We have a long way to go to answer this question. But right now, what we did in a paper that Sergei Nusdin was referring to was really kind of bite off the part of the question that's in this yellow box right here. How much seaweed carbon could we grow if we were to try to grow it in places in the ocean that could grow seaweed? Now, are we advocating that we should grow seaweed everywhere in the ocean? Absolutely not. This is more of like a back of the envelope first, check to see, could we get to climate relevant scales of seaweed growth if we tried, right? Then we should explore the next really important questions of should we do that and what other things would it affect? So we used the model to really take the first step, which is asking, okay, how much seaweed can we grow? And this work was led by a then postdoc of mine, but now a new professor at UCLA, Dr. Isabella Arzeno-Soltero. And she took our model and fed it with all these kind of vital rate growth characteristics of four different kinds of seaweed. So tropical and temperate red and brown seaweed species. And we planted each of those seaweeds everywhere over the global numerical ocean, and then saw how much seaweed we could produce using that. And what we saw was that the answer to that question is really dependent on what kinds of information we have about the nutrients in the upper ocean. And it turns out that the nutrients in the upper ocean are really is a hard thing to know, because it requires a lot of water samples, or we have some global models that model nutrients, but frankly, they don't agree with each other on how much nutrients there are. So I think there's a big question. The answer to this question we're finding is really sensitive to things that we still need to figure out, which is a good thing to know. So if you assume you have sort of the amount of nutrients that are in our global ocean, that all the seaweed can take up anything at the surface, then you get this kind of result on the left-hand side, which is a lot of seaweed. In fact, an unrealistically large amount of seaweed. Whereas if instead in our model, if we restrict the seaweed to take up only nutrients that are kind of mixed upwards from the deep ocean, sort of new nitrogen, then we get a much more conservative answer that is probably too small if you have just a couple of farms out there. So we're sort of bounding our answer here. But then we're also asking the next question, which is, okay, if you can grow this much seaweed, how much is it worth? And would it cost you more to farm it, say, in the middle of the Pacific, than it would to get the value out of the products, or the value out of the carbon sequestration? Would it be better to put investing your money into other forms of carbon sequestration than growing seaweed, say, in the middle of the Pacific? Mostly the answer is yes. But in order to make this kind of back-of-the-envelope estimate, you need to include all the costs of cultivating, like the farm costs, transportation out to the farm to harvest, the labor, and the value of the different products that you would make from the seaweed. And it turns out that sinking seaweed is, at least from these estimates, is usually not the most profitable thing to do with it, carbon-wise, that you could offset other types of intensive greenhouse gas emissions with seaweed products that are lower greenhouse gas emissions, like in food and in animal feed, than the value you could get from sinking that seaweed to the bottom of the ocean. And that's even before we think about the environmental implications of sinking a bunch of seaweed to the bottom of the ocean. So, this is a first step, but I think it's kind of, we are able to, we partnered with Carbon Plan to take our model and put it online, take a lot of the output so you can play around with it. If you go to Carbon Plan and you look up macroalgae, you can play around with some of this, this data output. All right. So that's it for me. If you guys have any questions, we have a couple of minutes. Oh, you know what? Not yet.
Hello.
All right.
Awesome. Thank you so much. This is super informative. I was super curious. You're saying that it doesn't make sense for profits to be able to sink it down versus other alternatives like food for people, farm foods, and we were talking about in the other tent over there that if you were to do that, that the carbon isn't actually sequestered because it gets utilized and put out into the environment again.
Right.
So talking about, I guess, did you have a good sense of like how much carbon could we sink to the bottom of the ocean? And
Sure.
I would love to understand a bit better, like what are some implications of that? How scary of a future are we talking about if we were to do that?
Yeah, good question. So according to our estimates, we could produce about a gigaton of seaweed carbon per year in an area that's twice the size of California. So we want to get to something like our targets for, if you look at like IPCC, for keeping our warming to 1.5 or 2 degrees, there's something like 15-ish gigatons per year. So 1 gigaton per year is a significant slice of that, right? So that's a climate relevant amount. But twice the size of California
is a huge, and that is twice the size of California in the most productive parts of global EEZs. And so that's a huge scale up. And that's before we sink it and get some fraction of that carbon sequestered. As far as the implications of sinking it and what's going to happen, we really don't, we don't have a lot of analogs for that much biomass on the bottom of the ocean. Whale falls, that's one that people are trying to look at to understand, okay, what happens when you just have this huge perturbation of a lot of biomass at the bottom. You know, I think we're thinking about, people are doing experiments right now, sinking terrestrial matter down and seeing what happens. I know some folks at UC Santa Barbara who have gotten some funding to actually sink seaweed down and study it. But turns out things happen, chemical processes on the bottom of the ocean, the deep ocean, happen really slowly. That's not where I usually work, but I was talking to an abyssal ocean scientist friend of mine, and they're like, yeah, sometimes we don't even revisit our sites for every 6 months, because things change so slowly out there that it doesn't make sense. So getting an answer to the question of exactly what's going to happen when we sink that much biomass is hard. But that's also why models are good, because we can accelerate our best guess to some of those questions.
Thank you.
All right, sure.
Why would you want to sink it? Can't you use it for other purposes, like I'm talking about?
Right. So, why you might want to sink it is because it's one way to get that carbon locked away from the upper ocean and the atmosphere. It's one way to kind of take it and put it in a longer, carbon cycle, farther away. So, but there might be other ways to do that. So, bioenergy with carbon capture and sequestration is one idea for, say, making energy from the biofuel in a process that also captures that carbon, and then you can put it in some geologic feature, right? So then you get some benefit from it, but you also have to find a place to put that carbon if you really want it to be a drawdown. Great. Okay, I'd like to introduce our next speaker. Right? Okay. So we have Armando Leon he's the CEO, co-founder and CEO of Algal Tech, and he is going to talk to us about control environment aeroponic systems.
Good afternoon. It's a pleasure to be here with you. My name is Armando Leon, and I'm CEO of Algal Tech. Algal Tech is a California corporation that has been doing work for many years. Let me give you some background of our company. This control environment, our aeroponic system, evolved from other business models that we have done through the years. This has been done by a group of scientists, engineers, entrepreneurs from the US and from Mexico. We've been working together for many years. Our technical team is led by Dr. Benjamin Moore. Ben is a plant physiologist from UC Berkeley. And in the 1990s, around 1995, he discovered a strain of a green seaweed, which is this one that I'm going to talk about. Is Urba clatrata. And he took this strain and took it to the University of Pennsylvania as a professor, and there he did the basic science and published a paper that he named Urba clatrata, a potential seawater irrigated crop. From there, I got hooked with him, okay? So then we started developing the business in Baja in the 1996, around there, and we worked together with universities in Mexico and research institutions from Mexico also in this program. So we have done 3 business models. The one I'm going to talk about is the third one. I'll tell you a little bit about the first and the second. The first business model was to grow Urba clatrata in Sinaloa, Mexico, in shrimp ponds that we're not using in the winter. So we will grow our strains of Urba clatrata and produce a ingredient for the feed for shrimp. And that was a very successful project from the point of view of scientific and research and development. not innovation, because we never were able to really put it in the market. The second business model, about 7, 8 years later, with Benjamin Mold came to me and said, hey, let's do something completely different. Let's do a co-culture of shrimp with Urba caltrata in the same pond so we can develop the shrimp. So we did that. And in that system, we developed the business in Sinaloa and in Baja with white shrimp and with brown shrimp. We got like 4 patents around the system. We sold the shrimp here in the US, but we were not able to sell our product, our second product, which is the Urba caltrata. So we had to stop the business, not because there wasn't any market, because as an entrepreneur and an ex-banker, we knew we had to cut it. We were losing money. So a few years later, Ben Mold called me again and said, hey, how about doing now just for consumption in humans, in nutraceuticals, also fresh? He said, let's talk about it. So we start Algaltech. And this is what I'm going to talk to you about. So as I told you, at Algatech, we developed this aeroponic control system. And because aonori can be grown in different forms, and by this, we discovered that this was the most productive way to do it. And the reason is because aonori has an impressive composition and can be used as a nutraceutical. Due to chronic diseases like diabetes and cancer and obesity, there is a growing consumer demand for potent natural ingredients like seaweeds to boost health. There's a great market demand and need for ingredients with superior functional properties, and there's also a growing demand from the industry for high-value ingredients that comes from seaweeds. Our solution is aonori. It's a new nutritious and health-promoting seaweed as a nutraceutical ingredient, which is a premium product for a premium market. Aonori has more nutritional and complete balance composition than any terrestrial vegetable. Aonori can be used as an ingredient in dry powder to boost nutrition in existing products or to design new products and add it to them. Aonori also acts as a flavor enhancement because it has a very light umami flavor. It's not a very strong umami taste, right? So it goes quite well with the North American palate. So that's why we had developed the system that you can see on the left here in the presentation, which is a system that is enclosed in a greenhouse environment, and the system is irrigated with salt water, and we use non-arable land. Okay? So the operation of the system is very, very efficient, and I'm going to tell you about that. First, I will tell you about the composition of Urba caltrata. Urba caltrata has a very rich composition. It's a package of nutrients. It has about 24% protein, and the amino acids that compose this protein are almost all what the humans need. It's very close to meat. Okay? It also has about 40% of fiber, total fiber, and about 23% of the total fiber is soluble fiber, or is fiber that can be used as a prebiotic. Okay? Then, again, the flavor of the product that we produce, because of the way we produce it and the freeze dry that we use to dry it, is a very mild umami flavor that can be very easily adapted to the North American palate. The other exciting composition that it has is the minerals, about 24% of minerals that you can see here, like iron, manganese, calcium, iodine. Okay? And there is also a package of vitamins, vitamin D, B12, C, E, K, and folic acid. So this is included in the package of nutrients. Additional, and is perhaps most important when we talk about nutraceuticals, is the fact that it has antioxidants. It has about 0.4% of lutein, which is a very potent antioxidant. Now, in our business model, we have developed our culture in Baja California, in Ensenada. With this small operation, pilot operation, we have been able to sample the market both in Mexico and the US. And we have now developed 4 products that we're introducing to the market. And these products are the seaweed by itself, aonori, as a high-value ingredient to be used as a nutraceutical product, to be used in many forms of as food or as a nutraceutical. It also, we also developed a product that is a capsule that is glutamine, 40% glutamine, and 60% aonori. Okay. The third product is a product that has collagen, 50% collagen and 50% aonori. And the fourth product is a green juice that has 50%, 60% aonori and 40% other mixes, and it has a great taste. It's a very palatable, very likable product. It's a green juice, and there is a lot of products there in the market that are green juices that offer many advantages for people to include them as part of the diet. But this will have a quite different composition, a much richer composition. Now, here I can show you the full system, the pilot program that we have in Baja. This system is basically an enclosed environment with a greenhouse, and the tanks that you see there is where we grow aonori. Aeroponics is a system to produce plants when you use spray water that serves to grow the vegetable and also to provide the nutrients. Okay, and it's done in a tank, so it's a recirculation system of the salt water from the ocean, and it's very efficient. It grows very rapidly, and because it's enclosed, it prevents fungi, bacteria, insects to come into the products. So the product that we are producing has optimal conditions. Here, in this slide, I showed some of the stages that we have for the production of aonori. The first one is we have the production in Petri dishes. That's where we keep our seeds or our strains. From there, we go to small tanks where we reproduce by vegetative propagation. And then we finally go to the operation in the full system. After 3 weeks, our crop is ready, and we harvest like if it was just grass, right? Just roll it up and take it to the processing location, and we have the products. Our system is very efficient. We use very little water, and we use salt water, and very low energy, about 24 watts per square meter, right? And it's sustainable, given that we don't use any fresh water or we don't use any arable land. And the water that we dispose is cleaner than the water that we bring from the ocean. It's clean by the seaweed, right? We can be away from the ocean. We can be a mile, 3 miles, 10 miles from the ocean, because we use very little water from the ocean, okay? And it's very healthy. We don't use any pesticides, herbicides, fungicides, nothing like that. And we have certifications, fully traceable, HACCP certification, and organic certification. Aonori is a seaweed that is very popular in Japan. There are 3 different species of ulva that are used. Ulva catrata is one of them. And the culture of aonori in Japan is done in the open ocean by fishermen that do fishing and growing the seaweeds some part of the year. And they dry it in the open air. And there is also a development by the University of Kochi that recently started a program to grow ulva caltrata in enclosed environments with hydroponic systems. Okay, so they are going towards that direction because in the ocean they're having a lot of trouble cultivating the seaweeds because of the climate change and because of contaminations in the waters, right? Okay, this is the last slide. We have here some samples of the way Japanese consume nori. They consume it as an organoleptic. In the center, you can see how it's mostly consumed in place of salt, right? Because of the monosodium glutamate that this product has, it acts to enhance the flavor. You don't have to use salt, so zero salt almost. Okay, and they also have other mixes like the ones you see here on the top, and there's also one that is very commonly used, mixed with other products. Okay. So this is the end of my presentations. Thank you very much.
We're going to have to hold questions for later. So I encourage you to contact Armando if you have some questions. I'd like to introduce the next speaker. Is Kalani here? All right. Next we have Kalani Ortiz who's going to tell us about bull kelp farming in Humboldt Bay. So just forward and back.
Or the clicker?
No, this is the clicker, yeah. So it'll control your slides.
And this is to go back.
Say again.
This is to go back.
That's right.
Hi everyone, I'm really happy to be here and get to talk with everybody, not just really about how bull kelp farming emerging really in Humboldt Bay, but a little bit about my project and how it was really engraved within this project. But before really going into a deeper dive on some questions and review of our farm in Humboldt Bay, I do want to go ahead and start with a land acknowledgement. So again, I want to begin by acknowledging that our seaweed farm resides on occupied ancestral homelands in the Wiyot people, which includes the Wiyot tribe, Bear River Rancheria, and Blue Lake Rancheria. So the Wiyot people have stewarded these lands and waters around Ouija, also known as Humboldt Bay, since time immemorial. The persistence of the Wiyot people remain in relationship with their territories despite their attempted genocide compels me to spread awareness to my community regarding the true history of this space. And as we continue to advance in these cultivation practices within aquaculture, it's imperative to center Indigenous voices in our planning and implementation to ensure that these steps taken to produce and mobilize knowledge into actions are representative of Indigenous community values and perspectives. And again, if you happen to be on Wiyot people's land, please consider contributing to the honor tax. And if you happen to reside on other ancestral lands, please feel free to look at Native Land California, which lays out general territories within our California coastlines. That being said, as I mentioned, went a little back. As we center to really cultivate and expand aquaculture within ancestral lands, it is important to look at also the crop species that have a potential cultural value, and one of them happens to be bull kelp. And again, bull kelp, since time immemorial, was used as a way of navigational pathways for a lot of tribes along the Pacific Northwest. But it's really interesting to also see kind of its importance along other factors, especially if we go into these main ones, aside from the cultural revitalization. So again, in an ecological importance aspect, since 2014, we've seen about 95% of the bull kelp canopies disappear from our waters due to the El Nino, or again, a blob that occurred heating our ocean waters and again affecting many of our foundational species of kelp. And in an economic potential, again, aquaculture has been also including applications of bulk kelp cultivation within Alaska and a lot of other areas within the Pacific Northwest. And a lot of these applications in the sense of long line cultivation, growing bulk kelp on open water, and also hatchery practices have now been centering into emergence of restoration applications as well. So it's a really exciting time in a sense of both combining economic and conservation strategies. But how do we really see, again, come back to the commercial perspective within the US and specifically in California with bulk kelp? So in the terms of the status of the industry within the US, again, we are still pretty, we're emerging in the US, but in California specifically, we're still considered underdeveloped. So again, the blue dots indicate the open water offshore farms that are currently located within those regions, along with the orange on the side also indicating the land base. So as we see, we are expanded in some areas, but if we're really accounting for the open water farms, the first open water commercial farm happened in 2020, and since 2021, 2 other have emerged. So just from that specific background within Sea Grant, that's a total of only 3 open water farms out of the 30 that are within US. And I'm happy and very grateful to be been able to conduct my research on one of the first. So my research is mainly centered off one of the first open water seaweed farms, again in Humboldt Bay, also known Weechee. And if for many of you that are not familiar with Humboldt, Humboldt Bay is located within Northern California, and it's an estuary that is ideal location for farming due to various mariculture species such as oysters and seaweed, along with environmental ideal parameters such as high nutrient content and being really a basis for coverage for a lot of species to really thrive in, specifically for seaweed. And so really, as I mentioned, my thesis really looked at many aspects of cultivation practices within hatchery and open water, but I do want to just give a general layout of our practices going from a nursery to the farm grow of bulk kelp to eventually harvesting our seed that we outplanted from our hatchery. So again, when we're discussing the nursery phase, and I'll go a little bit more into detail what that really looks like, but we mainly collected our bulk kelp from Trinidad Harbor out in Trinidad, California. And I'll kind of go a little bit of what specifically I collected within bulk kelp. And so when we're going into the nursery phase, there's really 4 components of collecting and processing bulk kelp in order for it to become a seed potentially and grow it out. So the first step, again, is collecting. And when we're collecting, we're, again, not collecting the whole individual, but rather the blades that contain something known as sori, or the reproductive structures of bulk kelp. And as we're moving towards that process of collection, we're really then stepping into the next phases of inoculation, incubation, which is really having those spores release out and have them attached to certain structures. In our cases, in a commercial setting, we really have them attached to spools, which are known as PVC wrapped with string twine, and then eventually going into the grow out. Oh, I think it froze. 1 minute. Okay. Oh, we're back on. Cool. Okay, so going back to the hatchery grow-out stage, as I mentioned, those spools are really what continue on in those hatchery grow-out stage. So as we're moving forward, we're really working with the system known as a flow-through system, or recirculating system, consisting of 8 tanks and 5 different spools in each of them. And I'll kind of go a little bit into what string type we're really using, because that's a big component of my project, or one of the questions within my project. And so really, with the nursery phase, it's really growing a lot of these bulk kelp early spores into their known as their sporophyte stage, which is usually the optimal stage of outplanting them from a smaller hatchery nursery setting to a farm setting for them to grow out to a mature size. But before going into that stage, what my project really consisted of is looking at 5 different stringing twines. So again, in a commercial setting, twines are usually the popular use mechanism for those spools attaching, but the big question is really understanding what twine to use. And so I'm going to go ahead and introduce 5 different ones that I use for my study. So the first one is known as a Gore-Tex twine. So again, it's a polyvinyl twine and a cotton fiber fused and creating a spacing between the lines. And again, you can kind of see that grow out within those weeks of stages. and then a Korean twine, and going along with even a biodegradable twine. And so again, our biodegradable twine is our hemp twine, then merging to our nylon twine, and again, a Japanese twine known as Kuramona. So as we're looking through these different stages of development, the first one, pre-inoculation, is really it not being inoculated at all with spores, and then moving down to week 4, where we start to see that grow out stage. And at week 8, we went ahead and started assessing these different twines before outplanting. And again, all of these spools reached about 2 millimeters in length of the sporophytes before outplanting. So again, in terms of the different twine materials, the assessment really consists of 5 factors. So again, it's percent coverage, the blade coverage, blade color, blade consistency, and minus the unwanted growth. So within that actual assessment, we were able to quantify within each spool that was in our replicate and understand which twine outperformed, at least in a nursery setting. And from our recent analysis, our Kuramona twine, which is that blue on the far left corner, found to have a high tendency within reaching that medium rate of 8.5, and actually was the one that outperformed the highest within a nursery. But again, this is mainly within a nursery setting. So in terms of the twine now being outplanted into Humboldt Bay, how does it do as it develops into its mature stage? So that leads us to now migrating of where our spools went to after. And so our farm site, which is where we started to grow, implement our outplant of the twine, is off the Samoa Channel, again in Humboldt County. So we're off the subtitle 2. And again, our farm is consisted of 2 farming systems, and I'll kind of go a little bit more into it. So our 2 farm systems are known as a single line and a multi-line system. Again, a single line is just one long line, which is 125 meters, anchored down at 2 ends, where a multi-line system is consisted of 4 to 5 lines that are actually on an array. So it's all being held by one bridle and consisting of having these 5 lines in order to promote higher biomass, but also be able to do that within a smaller area. So we now tested these 2, but again, the unique part of the multi-line system is we had it at a gradient. So the reason why we had it at a gradient, meaning it's coming from a shallower to deeper to shallower depth, is understanding also the development of these bull kelp within each of these sections. In natural settings, there's also there's been a lot of questioning between their development in deeper waters and how their morphological or physiological characteristics differ within these zones. And so part of that was really right now just focusing on what the harvest data really tells us between those systems. So again, going off of the data from our recent harvest back in June, we actually were able to grow up to 2,953 pounds of bull kelp. And again, as I mentioned, we did have depths considered in this, and I'm still in the process of analyzing. But as we seeded these lines within these twines within different depths, the future data will again tell us how the biomass differed within each those different depths, and how much either mortality or higher biomass we were able to harvest within those ranges. So really exciting time to be able to, just in general, grow bull kelp, but to even be able to grow it to that high biomass was just a really amazing experience. Along with this was really the experience of working so much with the Humboldt community. There was so many interests from many community members on really just alone the potential that bull kelp, once harvested, what we can use it or utilize it for, but also the interest of really learning a lot of these farming applications that we may have not been born or really grown into. So we worked with the Rue Gallagher Lab, Food Sovereignty Lab, which focuses on revitalizing a lot of cultural and indigenous food groups within Humboldt County. And their project worked with the bull kelp. They were able to come harvest majority of the bull kelp and actually incorporate it into this project known as the We Alder distribution program, creating food boxes for a lot of elders within the region. And then also working with local farmers and chefs who were using different applications of bull kelp, either as a fertilizer or again, even a salsa verde, which I have to say was probably my favorite salsa I've ever tried. And again, as I just want to thank again all the funders and supporters throughout this project, but really just thank our PIs. Again, I was the second grad student to really work on this experimental farm, and to even be on a farm in California in Humboldt County was an amazing experience. So thank you again to Dr. Rafael Cuevas Uribe, Rick Zeckman, and Karen Gray Geisler, who created this space for students to really experiment and expand their skills within the aquaculture in California. And again, thank you to all the volunteers, to all the organizations, and all the collaborators throughout this project. Again, this harvest took 3 days, so we would have not been able to do this without the help, again, of so many experts such as Sunken Seaweed, Hog Island Oyster Company's facility, and again, all the volunteers within my lab, along with also the university, and really make this a coalition project to really expand on aquaculture in California. So thank you so much for taking the time to listen to me.
Thanks for that talk. That was wonderful. Please come and ask our questions afterward. We have to move on to the next talk. So we have Carolina Pantano. Oh, great. All right. So Carolina is going to be, she's representing Por el Mar, and she's going to be telling us about kelp cultivation or kelp restoration in Patagonia.
Okay, thank you for that beautiful introduction, and thank you everyone for being here. I'm really nervous, but I'm so honored to be at the festival and having the opportunity to share a little bit the work of Por el Mar. So this video was to show you a little bit in images and not in words about our kelp forests down at the end of the world where I live. I don't know if everyone knows that in Patagonia there's kelp forests. Some people get surprised sometimes when like, you do have kelp forests there. And you have them here in California as well, and they're like in other parts of the world. So yeah, was to bring a little bit of that corner to you in images that are really beautiful to us. And obviously we had some divers there as well that are part of our team, which I'm gonna talk about our work a little bit now. So yeah, that's the presentation. Well, this is a beautiful presentation that our communication team made for me to share a little bit about our work. And they did this no to care lemma to like kind of know how important for us is to get to know our kelp forests and to get the people from Argentina also to know about this important ecosystem, to be able to like share and want to care about them in the different ways that we can preserve these forests in Argentina. Do I have to like change with this? Okay, great. Okay, so what is Por el Mar? Por el Mar is the organization that I work with in Patagonia, and we are a bunch of young activists, legal experts, scientists, and communicators that we have a common goal, which is obviously protecting the ocean. Right now we're working in Patagonia, but God knows who is going to take us later, where it's going to take us. So yeah, here, passionate individuals. I think that's like are the word that most described us, because we're all very passionate about what we do. So I was going to talk about, obviously, in the California Seaweed Festival, we have quite like 2 or 3 main projects where we work about, but the one about forests of Patagonia are is the most like important one to talk about here in the festival. So I'll be speaking about that project in particular. And before speaking about the project, I wanted to let you know about the current situation of kelp forests in Patagonia. So there are some works that already show that the southern distribution of kelp forests in Patagonia are like a global refugee for kelp forests, since are one of the least disturbed ecosystems in the world in terms of this kind of, in terms of this ecosystem. It is formed mainly by two species, which are Macrocystis and Lesonia, so giant kelp and Lesonia. These two species are distributed along the Patagonian coast from the state of Chubut, like northern Patagonia, till the southern tip of Argentina and Chile, obviously, because we share all this corridor with our Chilean brothers. So you can see that the kelp forest gets much more less seasonal, more stable, and the individuals get much bigger since when you go to like more southern waters, because they're richer in nutrients because of the Antarctic current, and obviously because we have colder waters. And the northern distribution forests are much more seasonal, and sometimes they've been like changing their distribution. So studying them and getting to know them better is our best like kind of weapon to like get to decision makers afterwards and let them know about not only the importance of this ecosystem, but also their threats and what tools can we develop to protect them and to protect the whole ecosystem. And we always say that we're kind of using the kelp forest as an umbrella species to also protect other species that depend on the kelp forest, and maybe we cannot protect them directly, but if we kind of like aim our efforts to protect the whole ecosystem, we'll maybe have indirect effects on other species as well. So that's why I wanted to show you a little bit about the underwater community around our kelp forests. So we have octopus, like a bunch of marine invertebrates live there. I don't have pictures of everything, but I'm going to tell you about all the associated species. This is the southern sea otter, which obviously feeds on sea urchins like the one you got here. This one is actually threatened in the island of Tierra del Fuego where it lives, but there's like no current efforts to work on restoration on this population. So we're kind of pushing for more studies on this species and obviously to work on maybe restoration and rewilding of this species in the south. Like sea stars, not all of the presentation is showing, but doesn't matter. The names are below, but it's not showing. So we have a lot of sea urchins species. We got some snails. We also got a lot of crabs associated with kelp forests. And we also have some cetaceans. Peel's dolphins feed on the areas of kelp forest beds all around Tierra del Fuego and Santa Cruz mainly, which are the two southern states in Argentina. So that is another species that we do not study, but there's a group that studies it, and we work along with them to like get to know a little bit about more their relationship and obviously how important the kelp forest is for this species of dolphin. So what does our project about kelp forest consists of? We call it the sea forests at the end of the world because obviously since Por el Mar has been working in the last tip of Argentina, which is called, the island of Tierra del Fuego. The tip of the island is called Peninsula Mitre, and Por el Mar started with its interest in kelp forest while working to become Peninsula Mitre a marine protected area and a terrestrial protected area as well. There we realized that this ecosystem was a very valuable one to work with, so we moved, since we had the successful protected area of Peninsula Mitre, we moved away to work with the whole ecosystem that was like developing there, which is the kelp. So we called it the end of the world. And Mainly, we work on 3 or 4 pillars to like kind of work on with kelp. So we do some not research just to make it like a university would do, like, and it's very important, but we try to develop the information necessary to the information necessary to share with decision makers. Then we also communicate a lot about this ecosystem to make community and the whole people know about the importance. So we share a lot of like underwater material that we do in our expeditions and everything to make people know about it. We also work with legal experts that obviously help us to know which are the best ways of approaching decision makers and get the tools for kelp forest conservation. And then we work also with local communities to get them to know and get them like engaged in our mission to protect kelp forests. I'm going to show a little bit. So here is kind of a sum up: explore, protect, and restore kelp forests along the coasts. Right now, kelp forests we don't really know if they need restoration. Oh, this is not looking entirely. Can we correct maybe this? Okay. I'm going to talk about a little bit what we do in our like kind of, yeah, great. Thank you so much. So when we gather information, it's very good to do it with different partners and obviously with different researchers that monitor the ecosystem in a long-term way and obviously in different levels. So one of the things that we're doing is trying to map through satellite imagery where the kelp was historically in Argentina, because there's no information of that, and where the kelp is right now. So what's the distribution? How much is it protected? What percentage? How are the kelp beds growing? Is their distribution changing? And we're finding, like, we just got these maps from our kelp watch partners that they're giving us the information and helping us like analyze this in a better way. So I just brought here ABC are 3 protected areas where we work. There are 3 marine protected areas. So we're kind of gathering information inside them. And you can see that, for example, in C, the most, like here, you have a big kelp bed here, and it's not protected, for example. And the marine protected area is just outside. So that's the kind of things that we want to punch out, like, hey, you're not protecting the biggest kelp bed, maybe. So that's why are we using maybe satellite imagery. And obviously we really want to like start comparing it with different environmental variables like sea temperature and everything. Like you guys here have the forest really affected by global warming, and we're not having that much. But yet, I know we're just behind in time, but it's just a matter of time till we get that effect. So yeah, it's just a matter of getting forward. Yeah, and this is like the Peninsula Mitre area that I was telling you about, Staten Island. I wanted just to show you where the kelp was around Patagonia. So we also do scientific expeditions to all of these different distribution areas of kelp. And that not only allows us to do some research, but also allows us to like obviously get some material to show the importance of this kelp forest. So we go with underwater photographers and we document a lot of this ecosystem. And here are some pictures of the things that we do with local communities. We give some lectures at local schools. We take the small kids also to look at the kelp, not only on the coast, but we're starting our snorkeling program as well. Argentina is not a place that looks a lot to the ocean. It's just weird because we have such an immense ocean just right next to us, but it's just the way it is. So we're trying with different strategies to like make people go into the ocean much more. Obviously, conditions are much more harsh than here, so it is understandable, but well, I'm almost finished. So, and there's another part that we're starting to explore, which is maybe thinking about like how can we solve the threats in another way, like thinking about more not prohibiting, not only like looking at marine protected areas or just declaring or protecting the species, but also like including human being in the solution and maybe think about nature-based solutions. That's when we started looking at regenerative ocean farming, which is something that here is a big thing and there's a lot of people doing it, but in Argentina it's not. And South America, maybe Chile is doing a lot of that, but very little. So we started exploring this, and we are thinking about doing a scientific pilot or a research pilot for regenerative ocean farming to see the impacts of this activity, the feasibility of doing it in Patagonian waters as well, obviously. So yeah, we're starting to like speak with the right people. For example, the people from Green Wave that are really experts in the matter, and that they're going to help us maybe to start this and in a small scale, and we just want to know how it would go. And we know that it has the potential to be a great option for Patagonian small towns, which are mainly dedicated to industrial fishing, but not even artisanal fishing. So this could be a good way to connect people to the ocean and also give them a solution for their economy and thinking about like local and small scale economy. So this is the message that we want to like let for you guys. When we unite, we have the strength of an ocean to protect what is vital for the planet. So thank you very much. Sorry if I stepped out.
Thanks, Carolina. Okay, we have up next Emma Akbajan, and she's going to tell us about UCLA kelp culture.
Fantastic. All right. Before I get started, can I see with a show of hands how many have you been to a kelp culture talk? Oh, amazing. Okay, that was more than I anticipated. And I have to say, I think I'm in the right group, being that so many of us are seaweed enthusiasts. Am I right? My name is Emma Ekmektion, and I'm a part of the kelp culture team at UCLA. We're an interdisciplinary community hosted by UCLA, and we put on monthly talks from people, diverse members of the community, all talking about kelp ecology and the future of kelp forests from many different perspectives. Our team consists of 3 people. Well, it did formally. Stephen Buter and I both started the kelp culture talks as a continuation and expansion of the Kelp Magic Talks that started in 2021 by Maura Palacios, who is a grad student at UCLA. Right now, it is co-managed by me and Jess Carson's Cass in the back. Shout out to you. And we decide themes for our talks and like to continue the community and create a platform where people can meet regularly to talk about kelp. Background about myself. So I actually don't come from a background in science, even though I have been independently researching marine ecology for some time now. I actually have a background in art, in fine arts, and my passion for kelp began in 2018 when I started going out to the Santa Rosa Island Research Station, an incredible place in the Northern Channel Islands, if you haven't been before. And that was a part of my undergrad experience when I was helping scientists clean waste off of the beach. And these are desolate beaches that nobody has seen all year except for the pinnipeds and a couple humans. And believe it or not, in just one weekend, we could get thousands of pounds of derelict fishing gear that have only been carried out by 5 people and crossed the ocean to be disposed of properly. And so I thought, well, I guess I'll use it as an art material. And that's when my love of art and science really got cemented. And I started to create these large-scale installations woven in the patterns of marine data collected by scientists for a NOAA marine debris grant. Most recently, I did my MFA from UCLA in the Design Media Arts Department, and that's where I met Dr. Siobhan Braybrook in the back in the hat, way to call you out, from the Braybrook Lab, who is the leader for Kelp Magic Group at UCLA. For my thesis project, I started doing some lab experiments in the Braybrook Lab. And the question that we had together was, can kelp adapt quickly enough to the changes that are happening in the environment? It's evolved over 180 80 million years, but suddenly the temperatures are going up, toxicity is affecting it. And so what we did is we extracted the heat shock protein, and we got data from Dr. Knudsen and Madeline Harden at the Knudsen lab. And we also referenced NCBI and realizing that there's just really not enough genetic information about kelp. And we still have so much to learn about kelp, as is proof by this festival. The result of this collaboration with the Braybrook Lab was a multimedia interactive installation where you could play with the DNA and see the difference between the DNA of kelp in Peru, both being macrocystis, and the kelp in California collected by the Knudsen lab. And this was just trying to push the boundary of how can we use science as a medium for art and art as a medium for science. So the 300 pounds of sea urchins that fit very unneatly in my car were gifted to me by Tristan Mayhew from the Nature Conservancy, and they're doing a really large scale restoration project up in Mendocino. But where does the kelp magic begin? Kelp culture is a result of the Kelp Magic Group. It's a project within it, and it's led by interdisciplinary professors Dr. Kyle Kavanaugh, Dr. Siobhan Braybrook, who are both on my thesis committee, Dr. Tim Malloy, who I've had the pleasure of working with for the last 2 years, who's a law professor, and many other established and interdisciplinary professors that are very interesting in their research. I want to talk about 3 sections to the Kelp Magic community at UCLA. The first one is the work in the Braybrook Lab. So Jess Carsons-Cass, who's in the back again, keeps giving a shout out to you. She's a PhD student, also co-managing kelp culture, and she's doing research on kelp microbiome, so swabbing blades of kelp and getting to know who's living with kelp and who's benefiting kelp. Former postdoc in the Braybrook Lab, Stephen Buter, was doing research on gene editing brown algae and creating biodiverse banks of sori for future restoration. So that's happening in the Braybrook Lab. Additionally, we have research that's been put on by Tim Malloy on the anticipatory governance side. So in the spring, we did over a dozen interviews with people like yourselves that we've found who have been working with kelp over the years. And we asked them in 3 situations, climate, restoration, and aquaculture, how would you feel about using biotech or genetic selection or genomic editing? And so we gauged their interest or their concern. And there was a lot of really interesting patterns like, oh, well, the general public doesn't support genetic modification, or they're scared by it because they don't know it, or, one of the biggest things, too, is restoration is really not a one solution fix. We really need tons of researchers and everybody on the ground helping the kelp forests. For those that we didn't reach in the interview, we also, Tim Malloy wrote a survey that went public last month, and so that was written also by Hannah Reed and Madeline Siegel, students at UCLA. And the survey asks the same questions, but to the public. And so I had the task of doing some science communication and illustrating the nuances between genetic selection or genomic editing to the general public to really see how people feel about restoration, aquaculture, just anybody. You guys could be taking the survey if you're on Qualtrics. You never know. Let's get into the nitty-gritty of kelp culture. So we started in fall 2022, and each quarter we have different themes for the talks. In winter 2022, or in fall 2022, it was art, biotech, and policy in the forest. So we had Josie, we had Taylor Griffith, who's here today, Dr. Kane Layton, and Robin Craig. So we wanted to get a really diverse perspective and overview of what people are thinking about when they think kelp forests from their respective disciplines. In winter 2023, we homed in on human culture and the forests. So I want to give a shout out to Alaka Alliance. If you're not familiar with them, they're a tribal-run organization that's working to reintroduce sea otters to the coast of Oregon to future-proof bull kelp forests. So that was Bob Bailey who gave a talk about that. We also had David Stuntaford with an industrial history of uses of kelp. And we had Lori Lewa Thomas, who we actually found out about through the California Seaweed Festival, which has been a constant inspiration for the kelp culture talks. In summer 2023, shout out to Kendall Barbary, who's in the corner there, we had a series of talks about aquaculture, and that was our biggest series so far. There's a lot of interest and a lot of overlap in research, and a trend that's coming up today, and also it was in the talks, was people don't really just want to do aquaculture for the business side of it, but also for the restoration, and see how those two can be really closely intertwined, because it's really important. What are we doing now? Well, feel free to pull up your phones and scan this QR code, because we have a talk coming up November 28th with Raina Wienhoff. I don't know if you guys are familiar with her, but she works in Europe, and she studies kelp sex. So she studies male and female gametophytes, and she recently published an article talking about how male and female gametophytes find each other in the intestinal tract of snails. So without that ecological relationship, would kelp be regenerating as much and as frequently? So just these interesting relationships in the kelp forest that maybe we hadn't considered before. So I hope to see you guys there. Future direction for kelp culture. We're trying to expand the community and just get as many people as possible to talk about kelp. I feel like working with kelp over the last, 8 years, there's always somebody I come across in every art installation that comes up to me and says, well, I don't know what the kelp forest is. And I'm like, that's okay. I think there's a lot of people that don't know. It's a little stab to the heart, to be honest. But it's really good to be able to talk to people about kelp and talk to people about their marine ecology and how it affects their daily life in a way that they might not have considered otherwise. And so right now we're doing the micro mini series, and that spotlights a lot of the work that's going on in the Braybrook lab, thinking about the unseen and trying to get to know kelp on a more finite, tiny level. Then we want to dive into restoration series in the spring. And I'm also trying to pull inspiration from people that come to the talks who are maybe I don't really have a background in kelp or in marine ecology, but I think it's cool and I'm here. So Ray Soto has been a frequenter of the talks, and he'll be giving a panel discussion in the spring, actually, talking about the Marymount campus that UCLA purchased and how we could potentially turn that into indigenous campus and marine resource center, just to try to think about futures for spaces to learn more about the ocean and learn more about the people that care about the ocean. So that's my talk. Thank you so much for being here today. That's you can sign up to the mailing list. I've got your email if you did the RSVP, but for if you want all the information, you can sign up to the mailing list. Yeah.
We have time for a couple questions.
Oh, awesome. Cool.
For any of the speakers, if you want.
Yes.
Are the kelp talks online? Can you
Yes. Yeah, so if you go to kelpculture.com, there's an archive section where it has links to YouTube where you can see all the talks that we've had. You can also subscribe to our YouTube channel. Yeah. Any other questions? I could also spin it back. Can I spin it back to them? Yeah, it's good. Yeah, what would you guys want to hear more of when it comes to kelp, ocean, anything? What was that? Processing kelp. Oh, okay. So processing kelp, very cool. Like in aquaculture situation. Okay, very good. Nice. Thank you for that. Yes, in the back. Yeah, so they were saying that it would be interesting to hear from somebody that processes kelp as bioplastic, and it almost sounds like maybe a workshop would be a cool thing. Yeah, so somebody that's been working with kelp hands-on to specifically do plastic stuff. I agree, that's super cool. Yeah?
kelp and the effect on the fish, the environment, their relationship?
So a fish person. Yeah, we should definitely have a fish person. And thinking for the restoration series, maybe branching it out so we have a sunflower star person, a fish person, a sea otter person, just one of each person that's we got to restore it for this species or this reason, and talk about that kind of trophic. Yeah, that would be super interesting. I like that. Yeah. Oh yeah. Yes. Okay. So on kelpculture.com, there's a link to present your work or share your research on our website. So feel free to explore that. Or you can just come up to me after this talk and tell me you want to give a talk, and we'll make it happen.
Thank you so much, Emma. That was great. All right, and kind of helping us to end this with a bang, we've got Yuanji Guo, and he's going to talk about some of the seaweed aquaculture research at the Hub SeaWorld Research Institute.
Thank you. Okay, thank you, everybody. So my name is Yuanji Guo. I come from the Hub SeaWorld Research Institute. So my topic is seaweed aquaculture research at our institute. Let me introduce our institute. Our institute has 3 campuses. One in Mason Bay is headquarters and have a lot of labs. Second one located in Carlsbad is marine fish hatchery, and we have another one located in Melbourne in Florida, focusing on marine mammal research. In Hubs, we are focusing on integrating seaweed into marine culture systems to deal with the ecological approach to the future marine agriculture. Currently, there have 2 land-based agriculture systems. One is recirculating agriculture systems. Another one is integrated multi-trophic agriculture systems. In RAI systems, the nitrogen is converted to the gas, so the waste is not turned into money. But in the IMD systems, the nitrogen will assimilate into produce the byproduct, so the waste can turn into money. So integrating seaweed into the fed species culture systems can to establish the IMD system to optimize the waste utilization, can diversify seafood production, also can resist the climate change. Currently in Hubs, we are focusing on 4 species culture. One is green algae alwa. The second is red algae gracillera and Davalaria. And also we culture the brown algae macrocystis. And we focus on research on the tongue culture and MT systems. Also, we want to use them to feed the sea urchins and abalone. The red colors in species are our future research species, and the colors in red, the research, are our future research field, including the open sea, long line culture, also open-based MD systems. Also, we want to develop human food, or we develop the animal formula diet. Currently, we have already set up 3 models IMT systems. The first one is pretty simple, just the finfish with the seaweed. The sand filled seawater flew through the water seabass tanks, and effluents from the water seabass tanks enter into the seaweed tanks. Also, the seaweed tank can directly get seawater from the sand filled seawater. So that means the seaweed tank can directly from the sand filter seawater, also can from the effluent of the tanks. We did 3 trials for this simple system. The first one is our grading level. The second is our stocking density and tank, the seawater exchange rate. Third one, we test the water from the effluents of the tank. Another one, just from directly the sand-filled seawater. The results is good, so the seaweed can assimilate 100% nutrients produced by the water seabass tanks. And the protein of the awa can reach to over 25%, but the nutrients assimilated 100%. That means the nutrients are limited. So if we supply more nutrients, the performance of awa can improve. Also, in this system, we didn't integrated invertebrates into the combined culture system. So the producer solid waste we want to use the invertebrates added into the system to use them. The second model is our recirculating system combined with the IMT systems. We can call this system is IMTRAS. This system is a little bit complicated. So the water from the tank of the yellowtail enter through the drum filter into the one sump. And after heating, pump into 3 awa tanks and back to the yellowtail tanks. During this process, the nutrients are assimilated by the awa, so that means clear seawater. And the solid waste backwashed by drum filter gravity flow into 3 mussel tanks, and then into 3 water-seeking cuber tanks, and back to the sump, right? So like completely circulating. So the awa exhibited very well, but the growth rate and productivity limited by the temperature and the light intensity because we did experiments in the winter time. The winter is so low, kind of like 14 Celsius degree, so they grow slow. Also because the figure on the bottom show the photo period less than 11 around. So that means during the nighttime, the photosynthesis stopped and the pH decreased greatly, which resulted in the ammonia increased. So that means if we can load more awa, or we can extend the photo period, can increase the photosynthesis to use more nutrients. And because we use the 20 kilogram cubic meter of the yellowtail, they produce a lot of nutrients. So we should add more awa biomass, or we can extend the photo period to increase the photosynthesis to use more nutrients. The third model is our cascade, the seawater flow systems. We set up 2 stages of the seaweed culture tanks. This is cascade. So seawater flow through the tank of the wet sea bass. Wet sea bass produce nutrients, produce solid waste. And they both enter into invertebrates tank in the middle. And the sea cucumber on the bottom can eat the solid waste. Top of the sea urchin also produce the solid waste. Deposited onto bottom, sea cucumber also can eat them. And the seawater containing a lot of nutrients enter into 2 stages of the seaweed. Seaweed can assimilate them, produce a lot of biomass of the seaweed, and we can use them directly feed the sea urchin or abalone, kind of like a circling, right? So the results, we just got primary results, the seaweed awa and the Valerianeria, they grow very well. And the tissue, nitrogen, carbon, other components are pending. This video show our tumbled culture dolls. So would you please help me? How we culture them? Here. So we culture this species before the month of July because temperature is over 2021, not good. Because we use the biomass of seaweed to feed the sea urchin, looks like sea urchin increased, the gonad, the index increased within 8 weeks. Also, the grade of the sea urchin increased greatly. Also, the taste is normal because it tested by the local seaweed distributors. We also culture Gracilaria and Macrocystis in the tampon culture tanks. We plan to use them to feed purple sea urchin or use in our empty systems oncoming experiments. Because we have 2 cases, 3 or 2, I can't remember, but 2, one in Carlsbad, the coast cases, one in the Santa Catalina Islands. We want to use these 2 cases as our research base to conduct research on the sea-based MMT systems in the future, very near coming. So this research funded by the PSMC and the Builder Initiative. Also, we get a lot of support from many organizations and people. Thank you. Okay. The temperature we can control in our recirculating IMD systems, but in the cascade systems, just natural ambient seawater temperatures. Yeah. But you can control if you want. Yeah, we just beginning to culture microcysts, the saboteur. Yeah, yes, temper. They grow well now. Yeah, because now if monoculture of fatty species, they produce a lot of pollution into the, you will discharge a lot of polluted seawater into the open sea. So we want to construct the amine system to use the waste, and you will discharge the very clear water, because in the land base, if we separate the component, it's easy to control them. So that's why our purpose. Thank you.
Thank you to all of our speakers. Can we give one more round of applause? And thank you all for coming out and asking great questions and being interested. And that's it for this session. So go enjoy the rest of the festival.